Hi, welcome to this video where we're going to be doing a mind map or a kind of run through of the key points from OCR module 2.3, which is all about nucleic acids. So that's DNA and RNA. So the first thing you need to do, if you're one of my students, is to get the PowerPoint slide, which I've set for you on Teams, and print out the pictures. So here's a picture of what the kind of PowerPoint, the mind map's going to look like. This is if you happen to have access to an A3 printer. And I've also attached this slide, which contains enlarged versions of those pictures, which you should then cut out and kind of arrange in your book to complete your mind map. Now, before we even get started labeling this, this diagram, I want you just to pause the video and spend a few minutes just looking at each individual image and seeing if you can remember what those images show you. If you'd like to, you can write some notes down in pencil so you can rub them out and correct them later if you've gone wrong. And then when you're ready to proceed with my version of the mind map, come back to this video, okay? Okay, here we go with the mind map. So first of all, let's zoom in a little bit into this top left-hand corner and talk about what is the fundamental unit of the nucleic acid. So the fundamental unit of a nucleic acid is something called a nucleotide. So a nucleotide is the monomer that, that builds up to form the polymer of DNA or an RNA. So a nucleotide contains three pieces. We've got a phosphate group, which is this circular bit here. We've got a sugar, and let's actually call it what it properly is. It's a pentose sugar. And then we've got the base. So the phosphate group is always the same. You don't really need to know the structure, but I'll just draw it over here just in case you really want to kind of go into a bit more detail. It's pretty much like this. Double bond O, double bond O, and then it has an O there and an O there. So it's like that. And there's a negative charge, which kind of jumps around actually between, it's delocalized across the different oxygens there, so it's a negatively charged. That is something to bear in mind. So I'll actually put that there, negative. Now the pentose sugar, that is variable, so it can be one of two things. So in DNA, we've got deoxyribose. And in RNA, we've got ribose. Now remember, we've got a couple of different types of RNA. You may remember we've got stuff ribosomal RNA, which is found in a ribosome, and we've got messenger RNA, and also we've got tRNA. Okay, so in all those types of RNA, we've got ribose as the sugar. So one thing to note that DNA, this deoxyribose, it basically just contains one less oxygen. So it contains an sort of H on the ring instead of an OH, a hydroxyl group. Okay, so the ribose has got an OH and the deoxyribose has just got an H. So sometimes you might be able, you're not supposed to know the structures off by heart, but you might have two pentose sugars and it might say which is ribose and which is deoxyribose. The one with fewer oxygens is the deoxyribose. Okay. So that's a bit about the nucleotide. Now, the bases vary, okay? So let's zoom into the bases. This is important, okay? We've got two different types of bases, okay? The first type of base that we've got is purine. And the second type of bases that we've got are pyrimides. Okay, so what are purines like? Purines are double ring structures. So you don't really need to know the structure, but you should kind of have an overview. So I'm going to try and draw one here. We've got basically in that double ring structure, we've kind of got like a pentagonal ring attached to a kind of hexagonal ring, like that. So it kind of looks a bit like that, okay? And then we have some, oxygen or nitrogen containing groups. So we kind of form the hydrogen bonds kind of here to the other base, which would be over here. So pyrimidines are single ring structures. Oops. Single ring structures, that says. Written that a bit unclearly. And basically the pyrimidine is just a kind of is just a sort of hexagonal type structure, like that. And then we can form some hydrogen bonds to the side. Okay, so which are which? Okay, so A and G are the purines, and C and T are the pyrimidines. Now I've missed one. The other one which I'm going to replace, which I'll put in a different color, is uracil, because uracil, remember, U is in RNA, okay? Whereas T, this one, is in DNA. Now, one thing we didn't say about the bases, I'm going to try and write it on this angle here. Let's see if I can do this, is that they contain nitrogen. Okay, the bases contain nitrogen. That's important later on. We're going to talk about the measles and Stahl experiment, which involves radioactive nitrogen. So that radioactive nitrogen gets incorporated into the bases. That's the only molecule that contains nitrogen in DNA. Okay. One more thing to say about the nucleotides is that they are formed by condensation reactions. Okay, so here we have the three parts of the nucleotide, and there'd be a condensation reaction here between the phosphate and the sugar, and then there'd be a condensation reaction here between the sugar and the base. So this would produce the full nucleotide, and it would produce also plus 2 H2O, because there'd be one molecule of water produced here and one here. Okay, so those are the bases. Now, what about DNA? How do we put the bases together into the structure of DNA? So if we come down to this section of our mind map, now we can see how we put the individual nucleotides together in DNA. So first of all, let's just label the structure of DNA. So here we've got what's called the sugar phosphate backbone. And in here, sometimes these are called the rungs, so like rungs of a ladder, and that's where the bases are. So let's call that a base pair. Rungs of the ladder. Okay, so how is this structure made? If we zoom in and look down here, we can see. So let's label the components of the down here. So first of all, we've got a nucleotide. Okay, so this nucleotide here links down to this here. So this is exactly the same as this. This nucleotide happens to have the base C. This, as its DNA, this is deoxyribose. And this is the phosphate. Now here is a base pair. And let's talk about the base pair actually a little bit more up here. Okay, so let's analyze what a base pair is. Whoops, that's a bit big. Okay, right, so base pair. It's always a purine plus a pyrimidine, okay, always. And that is because we always have a double ring structure paired with a single ring structure, so that the rungs of the ladder are always the same length. So rungs, the same length. So the pairings that we have there are A to T. Here we can see an A to T. Now this is actually a slightly incorrect diagram. So I'm going to put A to T, and I'm going to do it like this, okay, with two little arrows. Because there's two hydrogen bonds between A and T. Or it could be U, okay, so A to T or U. And then we have C, running out of space here, to G. Okay, with three hydrogen bonds between C and G. Okay, so this is actually correct. If we zoom in a little bit more, I'm going to try and correct this diagram here. So C to G, yes, I've got 1, 2, 3 hydrogen bonds. A to T, it's only 2, so I'll highlight those 2, and I will use a white here to just erase that one. Okay, so those are hydrogen bonds in the middle. Let's label those. They're hydrogen bonds. Okay, and let's say that they are between, we call them complementary base pairs. That's keyword complementary base pairs. Okay, and the rule is like that always. Okay, so A always binds to T or U, and C always binds to G. Okay, remember in DNA it's A to T, U, uracil is just for RNA. Now, one other thing, if I zoom out a little, one other thing that's important is you might have noticed these numbers here, 5, 3, 5, and 3. What on earth are they about? Well, they actually refer to this pentose sugar here. So the pentose sugar, you don't need to know much about its structure. Zoom in a tad more. The pentose sugar, a bit like glucose, it has an oxygen in the ring, and then we go round the ring and we number the carbons like so. So this would be carbon 1, this would be carbon 2, this would be carbon 3, this would be carbon 4, and actually carbon 5 is right here, halfway between the phosphate group. Okay, there. So when we're talking about the bonds, we can say that there is a bond between carbon 5 here to the phosphate, and down here there'd be a bond from carbon 3 down to the phosphate below. So that's what this refers to. This 3 is really talking about, if I just zoom in a little bit more, this 3 is talking about this specific point on the pentose sugar, and this 5 is actually really talking about this specific point, this carbon that sticks up there. Okay, so when we talk about the strands, we say that this one is going 3 to 5, whereas the other one is going 5 to 3. So the two strands, we say, run in an anti-parallel. Okay, because they're parallel, but one's flipped going the other direction. So they run in an anti-parallel fashion. And we can also say, put a comma there, and they are twisted. Into a helix, into a double helix, okay? Really, a lot later on at university, you also know that the helix is slightly asymmetrical because we have something called a major groove and a minor groove. I'll just highlight that as well. Not really on the A-level syllabus so much, but it's worth kind of just noting about. I'll put that in a sort of different color here. Yeah, that'll do. So this is called the major, groove. It's a bit too heavy there. And this here would be the minor groove. Now the significance of that is that most proteins that interact with DNA kind of slot into the major groove, and in doing so, when they kind of nestle into that groove, they can actually interact with specific sequences of bases in the DNA. So proteins can kind of bind only to certain sequences of the DNA. That will be important when we're looking at transcription factors and things like that later on. Okay, so let's zoom out. and a little bit further to our completed mind map. Well, that first part of the mind map. So what I'd like you to do now is try and turn that into a little paragraph. Just pause the video and I'd like you to describe the structure of DNA here. Okay, now there might not be full of space to write it out in sort of longhand, but potentially what you could do is you could take another piece of paper and just cover up the mind map which we filled in so far and see if you can just write down bullet point notes for an essay question. So if an essay question was describe the structure of DNA, 9 marks, could you put 9 bullet points down? Okay. Pause the video, write down your 9 bullet points in that box just to check that you remember and you can recall all of that information. Come back to me when you're done. Okay, so we've done this activity in class. You may remember we looked at an answer before. So here is the answer that we looked at in class, which I wrote. Okay, so this is a, hopefully I feel like this would be a 9-mark answer if this was a 9-mark question. So what you could do is just pause the video, have a read through that, and sort of maybe on your hand you can sort of count how many points you think that this answer gets when you get to a key sentence or key phrase. Sorry, put a point on your hand like this and see how many you get to. Okay, do that now. All right, now it's my turn to do that as well. So I'm going to highlight what I think is going to be good. Okay, so polymer, yeah, that's one. So the monomers are nucleotides, that's one. So we've got 2 marks so far. Okay, we've got this pentose sugar, probably 3, phosphate group and base. Let's just give that one. Let's be a bit stingy. So condensation reactions. There's going to have another one there 4. So saying that water's formed. 2 strands of nucleotides running parallel. Okay, that's probably 5, but in opposite direction. The direction of the deoxyribose sugar being reversed. Maybe I could have even talked a little bit about the sort of change color there. Could have maybe added about 5' to 3' on the sort of sugar. Oops, that's not sugar, sorry. That's a pentose, isn't it? So it looks like that. Maybe I could have even talked about the 5 and the 3, for example. There. Okay, so how many marks? So I've lost track of how many marks I've got, but let's keep going. Maybe you haven't. We'll talk about the backbone. Didn't talk about, didn't label that on my map. Got to go back and label this, although there's a spelling mistake there. Should be phosphodiester bonds. Phosphodiester bonds. We're going to put that on my map in a second. Nitrogen bases facing inwards, sort of the ladder and rungs. Probably another mark there. And then I talked about this specific purine and pyrimidine pairing. And I've also talked about the hydrogen bonds and the numbers of hydrogen bonds as well, which is also a mark. And then I've actually gone on to talk about DNA replication. And then I've talked about this double helix structure. Okay, so did you include most of those points as bullet points in your little summary box? How did you do? Okay, right, let's go back and add that 1 point to the mind map, which I forgot to do, and then we're going to continue on. So the one thing I forgot to say was that there's a phosphodiester bond. So I'm going to zoom in a bit. And let's maybe just highlight this segment there. You know, I'm going to do a different color. So there's no mistaking it. Let's highlight it in pink. And let's just highlight this segment here. Okay, so it's that whole thing. From one sugar to the next, we call it a phosphodiester bond. And I'll keep the same roughly color on that so we can label it like that. Let's make it a bit smaller and I'll say phosphodiester bond. If you're wondering why diester, well actually technically there's one phosphoester bond from one sugar to the phosphate, and then there's another phosphoester bond from that phosphate to the next sugar. So considering the whole thing, that's a phosphodiester bond. But normally in exams, Either phosphoester or phosphodiester would be given credit. Phosphoddiester is more of a safe bet, however. Okay, right. Let's move on and talk about DNA replication. Right, on to DNA replication. Okay, so this whole box here is the sequence, kind of cartoon strip style, of the events of DNA replication. Okay, and we say that DNA replication is semi-conservative. We can sort of see, we'll see why that is later on. So let's just highlight this, nice and big. Okay, so DNA replication. So what are the steps of this process? Okay, so first of all, DNA starts as a double helix. And there's something called DNA gyrase. Untwists. Okay, so gyrase is an enzyme that untwists. You need to know the names of these enzymes, so I'll highlight that. DNA gyrase. Okay, next thing is that we have then the enzyme DNA helicase unzips the helix. And when it unzips, it breaks hydrogen bonds. Okay, remember, hydrogen bonds are not full covalent bonds. They're sort of interactions that are a bit weaker than, quite a lot weaker actually, sorry, than covalent bonds. So they can be broken, but that's temporary, and then they're going to reform, or other hydrogen bonds are going to reform later. Like I said, actually, so they could, those bases that have been separated by the helicase, they could just snap back together again. But to prevent that, something called single-stranded binding proteins, these SSB, I don't know if you can see that on yours, it's a bit blurry on my image here, but well, let's write out in full, single-stranded binding proteins attach to prevent, use a bit of a technical word here, re-annealing. So re-annealing is when the two strands would join back together. Kind of imagine them like magnets, almost just locking together like that. So that prevents re-annealing. So let's just highlight that, single-stranded binding proteins, or SSB proteins. Now we have a new enzyme come in. I'm going to change color because this is an important part. So here we have DNA polymerase. Okay, so it binds to the leading strand. So this here is going to be called the leading strand. I might go over slightly here. It binds to the leading strand. And what it does is it moves along the strand, forming covalent bonds between different nucleotides. So I'll link a video in the description where you can see this happening in real time. But if this is the sort of strand that's acting as a template, what happens is nucleotides floating around inside the nucleus fly in and stick to the right place. And then the enzyme kind of moves along, joining up those nucleotides along the strand. So a common mistake is people say the DNA polymerase forms hydrogen bonds between the nucleotides with the corresponding nucleotides, kind of joining the rungs of the ladder. That's not correct. DNA polymerase joins up the backbone. Okay, the nucleotides fly in just by hydrogen bonding. They kind of anneal as if by magic. They're kind of magnetically attracted, sort of. We're forming covalent bonds here, this enzyme. Okay, so I'm just going to add that here. DNA polymerase binds to leading strand, forms covalent phosphodiester bonds. Okay, now the other thing here, I'm going to have to pinch in a little bit more. Yeah, okay, let's just go over here. So the other thing here is that it's significant that it's moving. So this strand here, this is 3 prime, this is 5 prime. So when we say it out loud, this, and it's right here, 3, that's not 3 dash, it's 3 prime. Okay, so this, if I were to say this out loud, 3 prime to 5 prime is what I would say. Actually, let's not say that. This DNA polymerase enzyme is moving, I'll put it in black, is moving from 5 to 3 prime. So it's moving in this direction, 5 prime to 3 prime direction. So let's write that here, 5 prime to 3 prime direction. So I can write it here, DNA pol only goes in a 5 prime to 3 prime direction. Okay, DNA polymerase only goes in the 5 prime to 3 prime direction. Okay, well that is kind of a problem, because if it only goes that way, then how do we copy this strand? Well, it's copied in reverse. So what happens is this DNA polymerase enzyme here, let's say it would have bound over here, can you see where the pen is, and then it would move along this way. So it's going in the reverse direction. So we can say down here that the lagging strand, which is this one, lagging, this one over here is the leading. So the lagging strand is synthesized discontinuously. So that what that means is that on this side, if you look closely where this pen is, on this side, the DNA polymerase enzyme can just move on up. And as it moves on up, this here, this replication fork, that's this point here, this replication fork just carries on moving up. Imagine just like kind of unzipping, like if you've got like a jacket which has a zip at the top and the zip at the bottom, it's just like taking the zip from the bottom and just unzipping it so the jacket kind of separates like that. Okay, so the replication fork can keep going, but on the lagging strand, you can't do that. Okay, so this thing is going to run out of space to go. Okay, and then it's going to join up here, and then it's going to run out of space. So you get these little gaps. So like if another one bound up here, it would go down and it would synthesize this, joining up the bases like that. But then there's a gap here, and then the same thing would happen down here. This one would reach the end where it had already been built, and there'd be a gap. So ligase joins up the gaps. See, the DNA ligase closes the gaps in the backbone. Now you might say, why is it that you have to have this ligase that joins up the gap? Because after all, DNA ligase is forming a phosphodiester bond. DNA polymerase forms a phosphodiester bond. So what's the difference? Well, if you want to go more depth into that, DNA polymerase can make a phosphodiester bond in a 5' to 3' direction. However, the ligase can do it in the other direction, so 3' to 5'. So that's why you have to have a ligase enzyme. Okay, so this is kind of zooming out. So what we have here is eventually the replication forks join, and until basically the kind of replication forks go up and go down at the same time, and eventually once the whole chromosome has been copied, then the two new pieces of DNA separate, the two new DNA molecules separate. So we call that semi-conservative replication. So DNA rep is semi-conservative, I'll just write semi-cons, because each new molecule has, and I'll try and keep the color code as of here, we've got one old and one new molecule. Strand has, so one old plus one new strand. Okay, can you see that in the diagram there? So this is the old parent DNA molecule, and here are two new daughter molecules. Same code, but you can see one's orange stranded and the blue strand is the kind of newly synthesized strand. So you could highlight that if you haven't got color, highlight that, make sure that that is clear on this diagram. Okay, so that's DNA semi-conservative replication. We can just maybe say that the evidence for that, and I won't go into a great deal of detail, is the Michelson-Stahl experiment, which basically used heavy nitrogen. So you can go and research this because I don't want to go into too much detail on this, but basically I kind of had two test tubes like this. These are supposed to be test tubes, and they grew bacteria in heavy nitrogen, and they sort of isolated the DNA and they spun it down. Okay, and then they got this DNA band here showing that it was heavy. And then after one generation, they got this band showing that all the new DNA was kind of not quite as heavy. They synchronized the division of the cells so they could really, just after one exact round of copying, they could isolate the DNA again and see how heavy it was. And it was a bit less heavy. And then the second time the cells had a chance to copy, then they got these two bands. So they got a light DNA band and they got a intermediate band, okay? And there was no more heavy. So this data, we won't go into a lot of detail, but this data can only be conserved by semi-conservative replication because this is all the old DNA. This is the new DNA that contained one heavy and one light strand. If you want to add that detail, you can. So, oh gosh, where am I going to put it? So one heavy, one light strand, forming a sort of intermediate molecule. And then this band here would be two light strands, and this band here would be two heavy. Sorry, one heavy, one light. Okay, so that's the Michelson-Stahl experiment. That's the evidence for this. Okay, so that's DNA replication done. Now let's go into transcription. So stay with me, or if you'd like to, you can pause, go get a cup of tea, and come back when you're ready to tackle transcription and translation. Okay, so now we're going to look at transcription, and that's kind of part of the turning of information from DNA into protein. So there's actually something called the central dogma of biochemistry, which is a bit of a fancy way of saying how cells work. Oops. And that is, see this big, that DNA, whoops, makes RNA, makes protein. Okay, now for that first step, DNA into RNA, we have the process of transcription, which is shown below. And then the second stage is going to be translation, which is going to be shown sort of over here. This is all to do with translation, all this is to do with transcription. So transcription is when DNA happens, it turns into RNA, and it happens in the nucleus. And the main enzyme involved is this enzyme here, RNA polymerase. Okay, so what happens? Well, first of all, let's label the different areas. So this is a nuclear pore here. Number one. Number two is the nuclear envelope, which is, remember, is a double membrane surrounding the nucleus. This here is a messenger RNA strand. And this, right, this is called the template strand. And this is called the coding strand. Okay, so what happens? when a gene is ready to be used, it has to be transcribed. Now we're going to look at how the cell knows which genes to kind of use when later in year 13. But for now, there's certain factors that need to come together to trigger the transcription and the use of a gene. Okay, they're called transcription factors, but we'll look at them more in year 13. So when the gene is triggered to be used, these factors bind and the enzyme RNA polymerase binds. Okay, it kind of temporarily kind of pulls apart the double DNA strands. So there's this kind of bubble, transcription bubble, and then the protein kind of moves up that bubble. So if you look where I am here, the movement of DNA polymerase, imagine this whole thing here would just be moving that way, okay? Moving along and transcribing. So the direction of movement of the RNA polymerase is this way. And again, what's happening when it's doing that is it is joining up the backbone. Free nucleotides will just kind of fly in and anneal next to the template strand, and then the RNA polymerase actually joins the backbone. So I'll kind of do that. Let's just kind of color that in a little bit. So it's this thing. That is what is being synthesized by the RNA polymerase, the backbone. Okay, all of this, and this is the RNA that's been synthesized already that floats out, and it's a different color here because it's slightly different. It's messenger RNA. So let's remember that messenger RNA, it has ribose, the sugar, not deoxyribose, and it has uracil instead of thymine. Okay, so if you look at well, if we were to look at the sequence of bases here, you know, let's see, I have to work this out from the diagram here. I'm going to zoom in a little bit. So this looks like it's an A. So if I wrote the bases in here, that's an A, isn't it? Oh, what's that? That's a C. That's a C. That is a G. I'm working this out from the code up here. That's an A, and so on and so on. You could keep going. So that means that if this is an A down here, remember A binds with T, but this is an RNA, so this would be a U, okay? C binds with a G, and so on. Now you could complete this just down to these three to see if you remember the pairings. Have you done it yet? Okay, so that's just going to be a G and a C, and again, it's going to be a U there, okay? So the code here on this mRNA is the same as on the coding strand, okay? So that's why it's called the coding strand. It's the same except T's will be replaced with U's, okay? So let's just write that there. Code or sequence on the messenger RNA is the same as this, brackets, T swapped for U. Okay, that's why it's called the coding strand. Of course, I didn't actually label here. mRNA stands for messenger RNA. This M is for messenger. So it is the messenger. It travels out of the nucleus via the nuclear pore and it goes off to the ribosome, which is where translation occurs. So let's jump straight into translation. We move across here. So this is the messenger RNA. Let's keep my same color scheme here. I'll just color that in like that. Okay, so this is the messenger RNA strand. Here it's got a sequence here. Okay, so let's label it there. So when the messenger RNA gets to the ribosome, there is a molecule called the transfer RNA, or tRNA, that helps the code on the messenger RNA be read. So tRNA stands for transfer RNA. Now the tRNA is this shape. Don't draw this, but I'm just going to trace it for you so you can sort of follow it. It kind of coils around in a bit of a noodle. Oops. And it goes around like this. Okay, so this is the actual, you can do that if you want. That's the kind of shape of it. It's a bit like a letter T, hence it's called tRNA, one of the reasons. Now there's two key important points on that. So you can see this top three letters here are highlighted. This section here, this part of the tRNA is called the anti-codon. codon. And this bit down here is the amino acid binding site. So the anticodon here binds to a very important, well, binds to the messenger RNA codon. So the codon is 3 letters on the mRNA strand. So the 3 letters here, for example, if you can see this, it says UGG. Those UGG letters on the messenger RNA strand bind with ACC on the anticodon. Okay? So every single code, for every single codon on the messenger RNA, every single possible combination, and there's 64 different possible combinations. So here are all the codons. For every single codon on the messenger RNA, there is an anticodon that will match it. And if the codon here, let's say, is UGG, then there will be a specific amino acid attached. So here, I can actually work it out. So UGG, so I have to actually substitute this here because this is the DNA codon actually. So UGG, so UGG would be tryptophan, okay? So in this case, because I can actually see what the codon it's binding to, and this is the map of the codon possibilities. A couple more things about these codons and this sort of sequence, this system of taking the code on the messenger RNA and turning it into an amino acid. This code, the DNA code, is universal. So let's just add 2 points about the code. Universal. So that means that every single living thing that we know of uses the same code. Okay, so from plants to animals to bacteria to protozoa. Okay, everything, and that is really, really, really strong evidence that we all evolved from a single common ancestor, which was the first one to sort of have that code. So it's a universal code, but it's also degenerate. Okay, now degenerate means that there are, oops, that's kind of a bit funny. Degenerate means that there are multiple codes for one amino acid. For example, if you look on this little thing here, you'll see that leucine has loads of different codes for it. Okay, so that this whole box there, oops, so leucine has got 1, 2, 3, 4, 5, 6. There's 6 different possible codes that will give you the amino acid leucine, for example. Now you don't need to know that, it's just an example of what this word degenerate means. And that is because there are a total, if you do the maths of having 4 different letters and 3 of those letters together, there are 64 different possible codes, 64 codes, but there's only 20 alpha, that's amino acid, 20 amino acids. So that means that we can use, on average, each amino acid will have 3 or 4 different codes that code for it. Okay, so that's the points on the code. There are some codes which don't have an amino acid associated. These are the stop codons, these ones here. Okay, there are 3 stop codons. Okay, we're going to come to those in a minute. Right, so the process of translation is happening here. Final thing. Let's label it nice and big. Oops, no, probably too big. Translation. Okay, translation. Oh, I did blue this time. So translation happens at the ribosome. It doesn't really happen in the ribosome, at the ribosome. And remember the ribosome itself. A couple things about it. It's got 2 subunits, large and small, and it's made of two things made of protein and something called ribosomal RNA. And that is just a structural RNA. So RNA coiled up in a big kind of tight ball that gives the ribosome structure. And actually, if you want to link this to the work that we did on phylogenetic analysis, the 16S rRNA is part of the ribosome. So it's one of the universal pieces of cellular machinery that all living things have, and we can analyze it and compare the differences between rRNA in multiple different species and work out their evolutionary relationships. Okay, so let's go into this kind of sequence here of how things are done. Let's pinch out a little bit. Okay, so translation. First of all, let's label. So up here, we've got the large subunit. And then here is the small subunit. Actually, the first thing that happens is it's the small subunit binds to the messenger RNA, and then the large subunit flies in, okay? So let's just label that. So small subunit binds to mRNA, then the large. It's been a while since I've done this, but I believe there's something called a Shannon-Delgado sequence that allows the ribosome to bind to that bind to the messenger RNA. Okay, so once it's bound, then a tRNA comes in. So there's the first tRNA comes in, and you can notice that there's a complementary base pairing between this anticodon here and the codon here, if you look where my pen is. Okay, and then a second one comes in. Okay, so let's kind of resume our labeling over here. Two amino acids kind of float in, and complementary base pairing between anticodon, I'm going to write kind of in bullet point form here, anticodon and codon. Okay, so now, now there's a complementary base pairing going on here. You can label them, hydrogen bonds temporarily forming. And when those two amino acids are held next to each other, because the tRNAs are sort of nestling into the ribosome has two sites where the tRNAs can sort of sit lined up next to the messenger RNA. And when they're there, we get the formation of a peptide bond. So this is key. Okay, so that is a key point here. So peptide bond formed between two amino acids. And if you want to go into a little bit more detail, there's an enzyme in the large subunit that does this, and it's called peptidile. Oh, I had to remember that there. So it's called a peptidile transferase enzyme or activity. Peptidile transferase. Oh, sorry, my pen's being a bit odd here. There we go, peptidile transferase. The reason it's called a transferase is because what it does is it moves the peptide bond. There is already a peptide bond there between the amino acid and its tRNA, but then it just moves that bond from the tRNA, so the tRNA detaches to the next amino acid next to it. That's why it's called a transferase. So now we've got this peptide bond formed, and the ribosome moves along one. Let's go back to the back. So ribosome moves along. And a new tRNA comes in. Not just any tRNA, but the one that is the correct one, where we can have the complementary base pairing between the codon and the anticodon. Again, here we can see that complementary base pairing between this next tRNA with its amino acid attached. We get another peptide bond form, and it keeps moving on. So peptide bonds form, and ribosome moves along until we reach a codon where there is no appropriate tRNA to match it. There are 3 codons where there is not a tRNA that matches it, and those codons are actually called stop codons. So this is the stop codon here, and there isn't a tRNA, so the ribosome pauses. I don't know how long it pauses. It sits there for a minute thinking, oh, what should I do? And eventually, basically, the messenger RNA just kind of floats away from the ribosome. The ribosome is done translating, and now we have this sequence of amino acids, which is the polypeptide. Now, we looked at proteins last lesson. So we know that they have the primary structure, which is the sequence of amino acids, and then that polypeptide would coil up, forming secondary and tertiary structure and its unique shape. It's also worth noting that in this diagram, this polypeptide is only 1, 2, 3, 4, 5, 6, 7, 8 amino acids long, when in reality a polypeptide would be probably, possibly thousands of amino acids long. So that's it. That's everything that you need to know about OCR A Level Biology module, which one is it? It's 2.3 on nucleic acids. So very quick review. We've looked at the structure of DNA, how it's a polymer of nucleotides, differences between DNA and RNA we've also looked at. Then we looked at DNA replication, how the semi-conservative process occurs, and briefly touched on the evidence for it. That's the measles and Stahl experiment. We've then looked at the central dogma of biochemistry, which is basically how information gets transferred from the code in the DNA to the proteins, which are the things that do stuff in the cell. Enzymes is our next topic. So that central dogma is in 2 stages. First, we have transcription that happens inside the nucleus. RNA polymerase is the key enzyme. And then that messenger RNA comes out of the nucleus via a nucleopore, attaches to a ribosome where translation occurs. And that involves this tRNA molecule. Okay, so that's it. I hope that was useful. Thanks very much, and I'll see you next time.